Cool. All right. Well, let's jumpstart. We've got ourselves an awesome session here. Today we have William Brandler. He's a data scientist with a background in whole genome sequencing data and analytics. He's now focused on enabling scientists and engineers to build solutions that have immediate real-world impacts. We also have with us Karen Feng, who is a software engineer at Databricks, where she builds solutions for healthcare and life sciences. Before Databricks, she developed statistical algorithms for genomics at Princeton University. So if you don't know, we are here introducing GLOW, an open source toolkit for large-scale genomic analysis. We'd love you to ask questions. We're going to answer some of the questions in the Q&A panel. So please go ahead and prop them into the Q&A panel. If you want to chime in the chat, we'll try our best, but usually we'll want to do it in the Q&A panel so we can keep track of everything. So without further ado, let's switch over and start with William and Karen.
Hey, Danny, thanks for the introduction. So I'm going to go ahead and share my screen. Okay, so you should see introducing GLOW, the header page for this talk. So my name is William Brandler. I'm a solutions architect at Databricks, and I'm focused exclusively on genomics and healthcare and life sciences, working with, as Danny said, with engineers and scientists to help scale up genomics in industry. And we also have Karen Feng, who's going to do the second half of the talk, and she's a lead software engineer at Databricks as well, working on Project GLOW. So just to give a brief outline of the talk this morning, or this evening, or this afternoon, wherever in the world you are, I'm going to give a brief overview of the current state of the field of genomics and talk a little bit about how it's transitioning from academia into industry, and then focus specifically on how genomics can be used in drug discovery, and talk about how genomics has become a big data problem over the past 2 to 3 years. And then I'll pass over to Karen, who's going to talk about how GLOW solves problems with big genomics data. And we're going to talk about how as genomics is transitioning from academia to industry, there's a need to go from ad hoc analysis using command line bioinformatics tools to a more robust, scalable production data science. So bioinformatics is being brought into the data science world, and it's enabling data scientists in industry and data engineers to work on a really massive scale. We'll talk a little bit about the partnership between Databricks and the Regeneron Genetics Center, which birthed the Project GLOW. And then Karen's going to go into some details about the innovations that are in GLOW and talk about how those compare to what's come before in academia. Cool. So genomics is taking off in industry. And I thought I'd show this tweet to start with from Janne Ehrlich, who used to be a professor of computer science at Columbia University and has now left academia and moved into industry. He's the chief scientific officer of MyHeritage. And in this tweet, he's talking about a list of professors in academia, big name professors who have left academia over the past 2 to 3 years. So this tweet was in March 2019. And then some people have replied. So actually there's a reply from Adam Orton, who's at 23andMe, saying no regrets. And so what are people doing in industry? Well, there's 4 key areas right now. One is drug discovery, which we're going to talk about later. The second is agrogenomics, breeding of animals. The third is cancer genomics. So this is doing single cell genomics, so sequencing single cells of tumors and discovering how they evolve over time. And then also liquid biopsies. So this is diagnosis of cancer via the blood. So tumors will shed DNA into the blood, and you can actually extract that DNA from the blood and sequence it and detect cancer at very early stages. And this is going to be the future of cancer detection, instead of the more invasive actual biopsies where you have to cut the tumor out when it already exists at a large enough scale. Well, that's an option. So genomics, and then the final use case is reproductive health. So this is the prevention of severe chromosomal abnormalities, either in pre-implantation embryos or doing non-invasive genetic screening during pregnancy. So these are the 4 areas that are going to be transformative in industry. It's going to lead to, it's going to radically transform the healthcare system. And ultimately, the goal is for genomics to be fully integrated into clinical practice. This is going to take a little bit longer because there are many challenges of getting this into the existing healthcare system. But I just thought I'd point out a story. This is from 10 years ago, written by Francis Collins, who's the director of the National Institutes of Health. I believe he still is. And he wrote back in 2010, 10 years after the announcement of the human genome, he wrote, so it's now 20 years since the announcement of the human genome by Bill Clinton and Tony Blair at the White House. And he talked about this child, 6-year-old kid called Nick Volker. So Nick has an inflammatory intestinal disease, and it had an unknown cause. And his parents went on a 3-year diagnostic odyssey to try and find out what the cause was of their child's disorder. And in the meantime, he had to have over 100 surgeries. He's in and out of hospital all the time. And finally, after 3 years, the cause was identified using this new technology called exome sequencing. The first proof of concept for this was by Jason Jury at the University of Washington. And what they did was they were able to extract the protein coding bits of the genome. So this is about 2% of the genome, and sequence it using this technology called next generation sequencing, which is commercialized by the company Illumina. And what they did was they basically had a bioinformatics scientist stare at a computer screen for 3 months, because back then there were no methods for analyzing this data. So it's literally like staring at the data and trying to understand where there was a mutation that was causing this disorder. And eventually they did figure it out, and it was in the gene XIAP, which is on the X chromosome. And once that gene was known, it actually immediately pointed to a treatment, which was a bone marrow transplant. And after the bone marrow transplant, Nick recovered, and he no longer has to have surgeries to keep him healthy. So there are countless number of cases like Nick where it's costing the healthcare system an incalculable amount of money and time, and the amount of suffering that children and adults have who have rare genetic diseases. And actually 30% of people have a rare genetic disease or will present with a rare genetic disease during their lifetime. So this is a massive, massive problem. And the first step in solving it is understanding the causes and making diagnostics in clinical practice a routine function. But this is about perhaps another 10 years away from being routine. But it's ultimately this is what we're working towards. But first, we're going to talk today just about drug development. So, and because this is something that is really in motion right now in industry, and the reason for it is that 90% of experimental medicines fail during clinical trials. So the success rate is pretty low, and over $100 billion is spent by pharmaceutical research and development teams every year. And out of that, only 10 to 20 drugs are approved. But the drugs that are approved, when you look retrospectively, it turns out that they're twice as likely to be approved if they have genetic supporting evidence. So even just having the supporting genetic evidence can slash the amount of money you have to spend on R&D, which is going to reduce the cost of developing drugs, which ultimately will reduce the cost of to the consumer. So there's massive cost savings to be had. And this is partially motivated these massive genome sequencing projects that are going on right now. So a good example is the UK Biobank, where they have genetic data on 500,000 Britons, and not only genetic data, but they have paired clinical data. And in the UK, there's a National Health Service, and everyone's treated the same way, and it's tracked throughout their lifetime. So they have incredible data to pair with the genomic data. Other countries are jumping on board with this too. So there's the Genome Asia 100K project based out of Singapore, which is ramping up right now. And also pharmaceutical companies themselves are doing massive sequencing. So Regeneron, so I mentioned earlier, Nick Volker, who was one of the first cases of exome sequencing 10 years ago. So back then, maybe there'd been 100 exomes that have been sequenced in total. And now, just 10 years later, Regeneron just by themselves have sequenced 1 million. So there's been just incredible progress over the last 10 years. And pharmaceutical companies are now adopting this population scale genomics. And they're using it to build just drug discovery pipelines and to prioritize their R&D investments. And just to dive a little bit more into how they're actually using the data, the first thing is, if you know the exact population frequency of a genetic disorder, then you can prioritize your investments better. So, if a disorder is too rare, it's hard to justify the investment right now because drug development costs too much. But if you do know that there's a rare genetic disorder that's actually, that may have variable presentation, so some people may be more severely affected than others, but they all have the same mutation. And if you can understand what the frequency of that mutation is, then you know whether or not it's worth investing in 2020. The second thing is the relationship, better understanding the relationship between clinical data and genetics. So there are many disorders that present in the same way, and you may give patients the same treatment, but when you look at the underlying genetics, there's actually different causes for that presentation, and actually different treatments may be appropriate. So this is going to increase the success rate of clinical trials if you can actually pair the genetics to the clinical data. And related to this is understanding how patients respond in clinical trials based on their genetics. So many of those 90% of clinical trials that fail, they may fail because a small subset of the patients have an adverse reaction. And the reaction is so severe, the side effects are so severe that it doesn't justify approving the drug. But when you can look at the genetic data, then you know that it actually is a subset of patients have these specific genetic mutations, and that makes them have the side effects. So if you can siphon those patients off and not give them the treatment, but give the patients that will benefit from the treatment, give it to them, then you can increase the likelihood of drugs being approved and actually reduce the amount of side effects that people have when they actually take the drugs. And then finally, actually, just using genetic data can increase your drug discovery pipeline. So you can increase the funnel coming in. So you can better understanding the causes of disease can point to new treatments. So just in conclusion, you get a bigger pipeline, so more potential drug targets, increase the likelihood of drug approval. So again, these things are increasing revenue and also reducing cost. So it's going to radically improve the efficiency of pharmaceutical research and development, and that's ongoing right now. So just to talk a little bit about Regeneron, who we've worked with for the past 3 years. They've set up their own branch of the company that just focuses on genetics, and they have the Regeneron Genetics Center, and they are mining the world's genetics data to extract insights. And so they sequence data themselves, but they also ingest data from public data sources. And they have collected data from various different cohorts of individuals with different diseases. So they have over 70 cohorts that they've collected. They have paired clinical data, for example, electronic medical records. And all they're trying to do is run association tests. So what mutations are associated with what diseases, and then they mine those results to get actionable insights. So in principle, it's a very simple problem to solve. But it turns out once you get to a certain scale, it becomes extremely complicated to gain these novel insights. And the reasons are threefold, and we're going to talk in more detail about this later, or Karen's going to talk a little bit more about this. First, it's hard to scale. So the data is decentralized in the cloud, and also all the existing bioinformatics tools assume a single node architecture. So basically everything was designed to run on a single node, or you can think about it like just run on your laptop. That's how all the bioinformatics tools have been developed. And that doesn't work once you get to hundreds of thousands of exomes or tens of thousands of genomes. So, and then secondly, the data is organized in different ways. So all these different cohorts have been treated in different ways. So it's a massive data engineering task to harmonize all of that data. And thirdly, because of the first two problems, it's just hard to get to insights because even asking simple questions just takes way too long. So massive problems in industry when you get to a certain scale. And really it's just become a big data problem, genomics, over the past few years, and it's been driven by this just immense innovation in sequencing technology, which came out of the Human Genome Project. So a genome costs less than $1,000 today, and the amount of genetic data is just growing exponentially. You went from 100 exomes to 1 million exomes in 10 years. In the next 5 years, the amount of genetic data is projected to grow to 2,000 petabytes, or 2,000 to 4,000 petabytes. And just to put that in perspective, the scale of the problem we have here, by 2025, YouTube is predicted to produce about 1,000 to 2,000 petabytes per year. The entire field of astronomy, which is also a big data problem, is about 100 petabytes per year. And Twitter, 1 to 17 petabytes per year. So genomics has the potential to be some of the biggest data in the world, and it already is in many ways. So the data is growing extremely rapidly. The costs are dropping rapidly too, and it's basically got to the stage where the bottleneck is not really the interpretation of the data, it's actually processing the data itself and getting to the interpretation. So I'm going to pass over to Karen, who's going to do the second half of the talk, and then we'll end with Q&A. Karen, would you like to share your screen?
Yeah, I think you'll need to turn yours off though.
Okay.
All right, so thank you so much for that, William, for that introduction. I'm going to be going into some of the more in-depth nitty-gritty of why the existing tooling in the genomics universe doesn't scale well to when we're working with biobank scale data. So historically speaking, the amount of data that people are processing are up to thousands of human genomes. However, as we're starting to work with 500,000 and up to millions of genomes, the file formats which exist today tend to be flat files. They are sort of like tab separated files, if you're familiar with VCFs. And those simply don't scale well. They start exploding as the amount of data increases along both the sample dimension, or the number of individuals for which you have genotyped, as well as the number of variants where an individual varies across the entire genome. So to dive in a little bit deeper into that, I'm showing a forum post from BioStars, which is sort of like the Stack Overflow for bioinformaticians. And how a commonly asked question might be, how do you split these flat VCF files, which are like a TSV or the tab separated file, by chromosome? And each of the sites here are essentially just a row in this flat file where every single individual is a column appended to a set of previous ones, which represent some basic information about a genetic variant, where the individuals differ from the reference build or the kind of average human genome. And this, you would think, should be fairly straightforward. However, the community of bioinformatics has not completely unified on what are considered to be the best practices. For example, one command line tool that you might want to use is the Genome Analysis Toolkit out of the Broad Institute. And here's a command line script that was suggested in this forum. But it's not just the GATK. There are other tools, such as VCF tools, Tabix, which is an indexing tool, or even a Java library called SnipSift. And so there are a lot of command line tools which are meant to be compatible with each of these file formats. And to work with them, you might have to do this sort of splitting and recombination yourself manually, which makes data management extremely difficult. And when you consider the sheer number of tools that exist to process this data, it becomes very complex, especially as you consider how many file formats are actually out there. It's not just VCF. This is the website of a commonly used genomic tool called Plink. And you'll see that VCF is on the page. However, they have introduced their own file formats, such as BedBIM-FAM, and there are a larger variety of them listed here as well. And other tools are compatible with overlapping, but not completely unified set of genomic file formats. So for example, the University of California at Santa Cruz has their own supported set of file formats, as does the National Cancer Institute and the group that manages the 1,000 Genomes Project. And as you can imagine, when you're trying to process these file formats that are not completely unified, you spend a lot of time, turns out, wasting on transforming between different file formats. And unfortunately, the accepted wisdom for how to work with these incompatible file formats that don't scale well is not to work with Spark or these sorts of commonly used data science tools. It's to get really good at command line scripts. Bioinformaticians are probably some of the best awk, sed, and greppers out there. They also write Python and Perl scripts in order to perform these sorts of transformations. And to work with these sorts of big genomic data sets, we need to be able to leverage the commonly used tools within the data science ecosystem for big data. We have been working with the Regeneron Genetics Center, who have the biggest genomic data sets out there today, including 1 million exomes. At this scale, as you can guess, awk and sed start tipping over. And in collaboration with the RGC, we've developed Project GLOW, which is an open source software project so that individuals in the community who are starting to work with data at this scale, including the UK Biobank, can avoid these common issues and leverage the lessons that we've learned in order to access insights in their data more rapidly. So what exactly is GLOW? Well, GLOW essentially is meant to bring bioinformatics under the umbrella of data science so that we can leverage the power of existing industry scale tools. In particular, GLOW is built on top of Apache Spark and is meant to be compatible with other tools in the big data, machine learning, and data science ecosystems. We've designed GLOW to also be backwards compatible with existing tools and file formats, such as VCF, within the genomics ecosystem so that users can ingest from these flat genomic files to Spark data frames and perform ad hoc transformations on partitions of the ingested data so that we no longer have to perform this manual splitting and recombination of the data. In addition, we've optimized common data transformations and exposed them as single line APIs. Finally, it's important to us that GLOW is open source for the entire community so that we can enable researchers across both academia and industry. And to go into each of the different points that I've just covered, GLOW is built on top of Spark. This allows users to start from our flat genomic file formats, such as VCFs or the Plink files, and ingest them into Spark data frames. These can then be joined with 8 other data sources in order to gain more insights. For example, we can perform a cross-join with phenotypic data, which would be a trait or a disease of interest, covariates, which might represent population structure, as well as variant annotations, which might represent how a genomic variant impacts a gene and protein expression downstream. These can then be stored in semi-structured form in a Delta Lake. Then on this larger data frame, we can use standard tools and APIs that are commonly used in data science and machine learning, such as pandas or koalas, which augment Spark SQL's functions to make them compatible with pandas. These experiments can then be tracked with tools such as MLflow for the purpose of reproducibility. As Spark is built to be compatible with a large variety of languages, including Python, R, SQL, Scala, and Java, GLOW is designed to be accessible for users from their language of choice. The second point that I mentioned earlier is that GLOW is designed to be backwards compatible. This is not just for the file formats, but also for the tools that are commonly used. And I mentioned earlier, VCF files, plain file formats, bed, bin, fam, these can all be ingested into the data frames. And when I mentioned that we're backwards compatible with the tools, it means that we take the files, put them into data frames, which are partitioned into smaller chunks. And then each of them is run through a Spark worker using the flat file format, so that we can go from a data frame, say that was read in from a bgen, which is another file format, and write those out as rows for VCF, ingest them through a command line tool such as Sage, and then read that output back into a data frame on the other end. So this is what's called the pipe transformer. And it looks like under the hood, you're able to run these command line tools without worrying about how we're able to split and sort this sort of data. But we're not forcing users to only use their old command line tools either. In addition to using Spark SQL expressions, we've exposed common APIs for the genomic workloads that we know people want to run. For example, this might include variant data manipulation functions, such as performing quality control in order to remove samples or variants which don't pass a certain bar of quality. We also have liftover, so that a set of variants can be lifted from one reference build to another. And this is a common pre-processing step. We also have variant normalization, so that when we get variants from multiple data sources, we can unify them in the representation. And we also include, among other functions, multi-lelic variant splitting, so that we can go from one representation into a set of exploded variants on which we can perform, say, a genome-wide association study on our single nucleotide polymorphisms. And this is when we're integrating with different data sets, such as clinical data or phenotypic data. We can calculate associations between genetic variants and our trait of interest. All right, so that's the kind of basic overview for what GLOW has as its basic functionality. I'm now going to be giving a quick overview of GLOW WGR, or GLOW Whole Genome Regression, which we recently released in collaboration with the Regeneron Genetics Center. And this is part of the genome-wide association study function set that I described. So what is GLOW WGR? Well, GLOW WGR is designed to remove population structure and thereby remove spurious positive associations between variants in the genome and the disease of interest or trait of interest. The plots here are what's called Manhattan plots. On the x-axis, you have the location of a genetic variant on the set of chromosomes, and the y-axis represents the significance of the association between the variant and the trait of interest. And when you're removing population structure from the equation and thereby removing these positive associations that are actually not real, the Manhattan plots kind of get a little bit shorter. The variants that were previously associated and thereby were high up on the plot are kind of brought down a little bit. And you'll see here that the Manhattan plots that result from Regini, which is the single node version of GLOW WGR, when compared against other tools that are commonly used in research, such as FastGWA or BoltLMM, the Manhattan plots look very similar. And this demonstrates that we're accounting for population structure similarly. But where GLOW-WGR really shines is in its ability to scale to any sort of data set that exists out there today. We've been processing the UK Biobank data set, which includes 500,000 individuals today. And you'll see here that the runtime is significantly decreased. It's actually a 10x improvement over the state of the art, which we just released via Regeni with the Regeneron Genetics Center. And under the hood, if you want to nerd out about it a little bit, the reason why Regeni has such massive improvements over existing tooling, and GLOW-WGR can improve on that even more today, is that Regeni takes the massive matrix of the number of samples across the number of variants and blocks that into smaller chunks. And then we can process each of those chunks separately. This allows us to be much more efficient when it comes to memory consumption, as well as computational resources generally. And as you can imagine, when we perform this sort of blocking and running the processes in parallel, this is really designed to be run on top of Spark. And therefore, GLOW-WGR can really demonstrate significant improvements over the state of the art. So that's all I have for today. Thank you so much for listening. We're going to be going to Q&A after this, but if you're interested in learning about what we've been working on and in order to contribute to our project or just try it out, feel free to visit projectglow.io, and we look forward to seeing you on our GitHub.
Okay, thank you, Karen. So it seems like most of the questions have been answered, but if you have been answered live. So if you do have any further questions, just pop them in the Q&A. And I'd just like to point out that this is the first of a series of tech talks that we're going to have on GLOW. So this one's the first one, and just giving an introduction and overview of what we're doing here. And the next ones will have a bit of a deeper dive into how you actually use GLOW, including demonstrations of notebooks and tutorials. And we're going to talk about how you actually run these large-scale genome-wide association studies. We're going to talk a little bit about the origin story for this extremely innovative new methods for doing genome-wide association studies with Regeneron. And then we'll talk about how to actually go, from the GWAS results to some insights as well. So this series of talks will be coming out over the next few months. So watch out for them.
Cool. Oh, sorry for that. I forgot to turn on my video. There we go, better. So thanks very much to William and Karen for this great session. I did want to call out, I'm going to share my screen, and I did want to call out since we've actually answered most of the questions, but we really want you guys to engage with us. So we've actually got some more sessions, just like Will was talking about. So let me go ahead and present my screen, please. Give me 1 second. Cool. So as Karen had already noted, and William noted, if you have any questions, please go to the Project GLOW website, projectglow.io. Code, including notebooks, documentation, the forums and Slacks. We're actually pretty active on these forums. So please come on, join us and talk to us here, okay? So there's a question which is very appropriate. How do we get assistance? Well, join us in the Slack or join us in the forums, and we will be there to answer your questions. If you want to watch this session again, please go ahead and go to youtube.com/Databricks. It's actually recording on YouTube Live as we speak. So the video is going to be available right after this. So you can watch again, share it with other folks as well. We can go ahead. We're going to make sure we update the meetup with that information as well. Okay. All right. So one of the first GLOW sessions that one of the sessions, excuse me, not first ones, actually, one of the sessions for GLOW that we're going to be going ahead and presenting on Tuesday, September 8th at 9 a.m. Pacific. It hasn't been announced yet, but will be announced very soon, is, as noted here, is genome genomic variant data ingest and manipulation using GLOW. So please join us for that session. A couple other things to note, we actually will probably have another 2 GLOW sessions announced in the next 2 or 3 days. So do stay tuned for that because we will be chiming in here. Let's see what else we have here. Okay, well, now we're going to switch gears back to the data and AI online meetup. So not as interesting for you GLOW folks, but if just in case for the data and AI online meetup folks, the next session will be Delta Lake 0.7.0 plus Spark 3.0 AMA. So come with your questions. It'll be next Tuesday on August 18th at 9 a.m. It'll be with Barack, TD, and myself. You're going to ask us questions and hopefully we have some answers. Okay. And then the following week, we're going to go ahead and be Douglas Moore and myself. We'll be talking about generating surrogate keys for your data lakehouse with Spark SQL and Delta Lake. And then on the 27th, Thursday 27th, that's a typo there, but Thursday 27th at 9 a.m., we're going to have a tutorial on how Delta Lake supercharges your data lakes with Louis. So, and then finally, as a quick note, if you want to get your own complete copy of the Learning Spark second edition ebook, go to this link. I'm going to leave it up for the next couple minutes. Databricks.co Meetup Spark book. That's the entire book. Okay, so we're not giving away the chapter, we're giving away the entire ebook. So hopefully, as you can tell, we're going to have a lot of more GLOW and awesome data and AI online meetup sessions. So we'd love to see you join us, ask questions. If you have other questions that we couldn't answer right now, it's okay. Join us on YouTube and put your comments there as well. Or as noted earlier on, go to projectglow.io and boom, go ahead and join us in Slack or join us in the forums and ask your questions there. So I think that's about it for today. Karen, William, anything else you'd like to add? Otherwise than that, we're good to go, really.
Thank you very much for organizing this, Denny.
Yeah, thank you so much. Please drop by our Slack channel. We weren't able to answer all of your questions. Some of those a little more complex. So just come by and we'll help you with anything else you might want to know.
Perfect. Well, then, thanks very much. Appreciate the time. Oops, oh, there we go. Appreciate the time. Ooh, there we go. And thanks very much for joining us today. Take care.